very good morning and a warm welcome to all the participants of the SEAMEO Network and affiliated countries to IMR-SEAMEO Webinar Series 2024. The theme for today is Electron Microscopy, Institute for Medical Research. Myself, Shalini Mathialagan, a medical laboratory technologist, also from Electron Microscopy Unit, IMR, will be the moderator for today's session. Ladies and gentlemen, this webinar series was initiated with an aim with mind to share knowledge covering the electron microscopy of Unit IMR. Today, we have with us the head of unit, Electron Microscopy Unit, and a research officer who are about to share their experience and knowledge with us, which related to today's theme. Before we begin, please allow me to announce few housekeeping messages. We will be not taking any questions during the presentations, but participants may ask your questions through the link provided and also in the YouTube channels. Question and answer sessions will be conducted after the presentations. Attendance links will be provided for those who would like to receive an e-certificates. Lastly, please subscribe to our National Institute of Health YouTube channels to receive our upcoming webinars. We will be begin the sessions by introducing our first speaker, Puan Tengku Rogayah Tengku Rashid, a research officer and also the head of the unit. She holds a bachelor degree in zoology from the University of Malaya and also have a master's in entomology from the National University of Malaysia. She works in the entomology field for almost 4 years before being transferred to virology units. After spending nearly 19 years in virology, she lastly joined electron microscopy in 2023. Her research areas are infectious disease and molecular virology, especially dengue, Zika, chikungunya, and respiratory viruses such as influenza, RSV, Boca virus, SARS-CoV, and also MERS-CoV. I would like to call upon Puan Tengku Rogayah to deliver her talk on the application in biomedical research and diagnostic. Puan Tengku Rogayah, the screen is yours.
Thank you, Ms. Shalini, for the kind introductions. Assalamualaikum and very good morning, everyone. I am Tengku Abdul Rashid, Research Officer from Electron Microscopy Unit. First of all, I would like to thank the IMR-SEAMEO Webinar Secretariat for inviting us to present in today's sessions. So I will start my presentation with a brief introduction to our unit. The Electron Microscopy Unit, IMR, was established in 1980 under Departments of Parasitology, and we received the first equipment in 1981. And over the last 40 years, the equipment has been replaced a few times, and the latest was in 2017. So far, right now we have one unit of TM, one unit of SEM, and with the strength of manpower of 7 people, and all ladies. And as one of the unit under Pusat Sumber Khas or Special Resource Center, our functions are not only to conduct research and provide special diagnostic services to hospital. We also provide and support complement research. We also support and complement research activity in IMR, other institutions and university. And we also provide consultations and trainings on the applications of EM to improve and strengthen the research activity. So the idea of electron microscopy technology has a very long history, where the lens theory has started as early as in 1920s. However, the first electron microscope was invented in somewhere 1931. And after the significant improvement and advancement in the technology, the SEM and TM was invented in late 1930s and early 1940s. So what is transmission electron microscopy and what is scanning electron microscopy, and how does it work? So in this slide, I will present a very brief principle of both tool, because the next presenter will present the technical and principle in more detail. So transmission electron microscopy, or TEM, employ a high speed focus beam of electron, where this electron will transmit through the super tiny sample, where this electron will pass through the sample and interact with the atoms and structure of the sample, and we form the image in the detector. Okay, presented in the slide under the TM column was the image of negative stain rotavirus. As you can see, the background was stained dark, while the virus was show a clear spot against the dark background. And how about the scanning electron microscope? So scanning electron microscope, or SEM, use a high-speed focus beam of electron, also similar like TEM. However, instead of transmit the electron through the sample, the SEM will scan its surface. While it scans, the electron beam will interact with the atoms in the sample and will reflect, will be reflected, and some of the electron will be ionized in the samples. This emitted electron serve a signal to build up the final image, as you can see in the electron micrograph of E. coli, where the 3D image was resulted from the different reflected angles of the electron. So the electron microscopy applications was very wide and very versatile. Nanoscience and nanotechnology, of course. And for biological and biomedical research, the application is used to investigate the ultrastructures of cells, organelles, macromolecule, and to study the mini microorganisms such as virus, bacteria, and their morphology and their interaction with the whole cell. And the technology also very important in disease diagnosis, for example, the biopsy sample and biological sample. Okay, how electron microscopy contributes toward impactful research. So with the advancements in other technologies, such for example in molecular technology, for example in NGS, metagenomic, metatranscriptomics, or these omic revolutions, is this electron microscopy still relevant in biomedical research? So the keywords in the answer is the magnification and high resolution imaging, where these two character, or these two character, is very almost exclusive to the EM technology. With this technology, it can provide exceptionally detailed image, which can help the researcher to, for example, to visualize a structure at the atomic levels. With the scientists then gain insight into the disease mechanism, and this detailed image also facilitated the nanotechnological research, such as development of new nanoproduct, where the knowledge of their structure is very important to ensure both the safety and efficacy of the product, and characterize the spatial relationship between the nanoparticles and the cell surface. And it is also very important for rapid diagnosis of the pathogen via their morphology and their interactions with the host cell. So, In the next few slides, I will present some of the EM applications in research that have been carried out in the EM unit. Some is already completed and other ongoing, and some are just started. Okay, we receive various type of specimen in our departments, from human specimens, uh, cell culture, virus, insects. We also receive animal tissue, and bacteria, fungus, and once in a while we receive material samples. Okay, the electron microscopic technology was widely used in viral diagnosis and viral research, because viruses are generally too small for inspection by light microscope. And one of the important factor of EM is the rapid catch-all methods. So this rapid catch-all methods of EM became very crucial for the rapid identifications of pathogen, especially as an early diagnosis method or during the outbreak of unknown pathogens, and for classifications of the pathogen based on their morphology. And these visualizations of virus particle provide a direct image at the nanometer scales and unbiased of their genomic material, whether the DNA virus or RNA virus. Okay, EM was also widely used to study the interaction between the virus and the host cell, the biology of the virus within the cell, where this understanding is important in containing viral disseminations, vaccine development, and so on. So in the micrograph was the image of BHK, or baby hamster kidney cell line, which infected with the chikungunya virus. It show the locations of the virus replications, virus shedding, and how it behave in the cells. Okay, the EM techniques also crucial in renal biopsy, where renal biopsy is the gold standard for diagnosing of renal disease. In recent years, the EM technique has been widely used as a supplement with other existing methods, such as immunofluorescence and light microscope technique. So the imaging of renal biopsy tissue by EMs allow the assessment of ultrastructure changes in the sample, where this imaging can reveal the detail at the cellular and subcellular level and contribute significantly in diagnosis of the renal disease. For presented in the slide are the image of few renal biopsy symptoms, the nephrotic syndrome. The condition result from the excreting too much protein in urine due to kidney disorder. So you can see the subepithelial deposit are visible along the glomerular basement membrane, and in minimal changes disease, the micrograph show the extensive flattening of podocyte food processor. So, EM technology was widely utilized in vaccine development in few stages. For example, in virus visualizations and characterizations, it allowed the scientists to visualize virus particle directly, which it revealed their size, their shapes, the surface features, and aiding in vaccine design. And at the structural insight for antigen selection stage, the technology will help identify regions of the virus that has strong immune response, where this region become vaccine antigen, which stimulating the production of protective antibody. And finally, at the control, quality control and vaccine production, the TM will ensure the integrity and purity of the particle. It verify that the vaccine contain the desired viral component without the contaminants. The technology also used to study the effect of antibiotics to the pathogen. For example, in this study, the effect of antibiotic on the E. coli morphology with the title Structural and Morphological Studies on Variable Affecting E. coli with Selected Commercial Antibody. And from this study and from the electron micrograph image, the treatment sample show that rupture and uneven morphology as compared to the control specimens. In assessing muscle tissue damage, so EM complement traditional histological technique and contribute significantly to understanding muscle pathology. Uh, in assessing muscle damage, tissue damage, the micrograph allow the study of ultrastructural abnormalities and vascular assessment. It revealed the detailed changes within the muscle fiber, and a detailed image show also allow the doctors to evaluate the small blood vessels, which added in the differential diagnosis. Okay, lastly, I will present the applications of EM in analyzing insects' mini structure. The SEM, scanning electron microscopy, microphotography, are used mainly for documenting external features of the insect, where it provides more info on tiny surface structure of the insect, such as trachea, such as asexual, or such as tracheoles. And this research provide aid in the study of interaction between the insects, for example, the insects and the plant, where the image can reveal the detail of the plant sample, plant structure, and also the insect structure, for example, the mouthpart, antenna, and also others. So, in other words, EM provide a window into the hidden worlds of insects. Okay, in conclusions, EM is a highly advanced microscope that blasts electron into the specimens and transmit electrons or excite electron which make up the specimen image. And this ability of this microscope help us to visualize specimens that are ultra small. And in summary, EM significantly contribute to biomedical research by revealing the details and enabling in structural biology and cellular imaging. With that, I end my presentation. Thank you for listening, and I will pass back the screen to our moderator. Thank you again.
Thank you, Puan Tengku Rogayah, for this interesting talk. Without any further ado, I am pleased to announce our second speaker, Puan Nur Afrina Muhamad Hendri. She is a research officer in Electron Microscopy Unit and holds a bachelor degrees in applied physics from Michigan State University, USA. She has been with the EM unit since 2018, and her research focuses on ultrastructural mechanisms of gram-negative bacteria and the effects of antimicrobial substance on bacteria. She also works on a renal biopsy ultrastructural studies and has been involved in 15 research projects, serving as the primary investigator for 2 of them. I would like to call upon Puan Nur Afrina to deliver her talk on the applications and operations of the instrument used in electron microscopy. Nur Afrina, the screen is yours.
Hi everyone who's watching right now, and my name is Nur Afrina Muhamad Hendri. For today's presentation, I will focus more to the instrument in application and also the operations. Okay, as we all learned that about light microscopy, which was the standard in the middle of the 20th century, and it's remained tremendously useful until today. But as we begin to learn more about the microscopic world using light microscope that could visualize the individual cells, we started to become curious about the structures that are smaller than a cell, and also some details of which were difficult to visualize and understand through the light microscope. Fortunately, that's why electron microscopy was introduced. Okay. Some electron microscopy techniques employ a resolution of zero point 0.2 nanometers, which is more than 1,000 times better than light microscopy. Which the higher with the higher resolution, electron microscope are able to magnify their specimen up to 1 million times their original size, and allowing researchers to study proteins inside the cell and the molecular structures of viruses and also other things which led to a better understanding of the microscopic world. Okay, of course, we have to ask how they do this. Well, the answer, of course, but through its name, which is they use beam electrons, which interact with the sample as the beam transmitted through the specimen. As we recall in study of chemistry, electrons are stable subatomic particles that carry a negative charge, and electron beam have much shorter wavelength than a visible light that light microscope use, which we refer as de Broglie wavelength, as we learn in the physics series. And this shorter wavelength allows for a higher resolution of detail and the higher level in imaging. Okay, for electron microscopy analysis, sample preparation is the most crucial part in producing a great results. Which is that's why all the samples need to undergo for the proper preparation before we see under TEM. Okay, first is the fixation. Okay, chemical fixation involve killing and preserving the organism, organ, tissue, cells in as lifelike condition as possible. Our ability to observe fixed biological ultrastructure using EM allow us to infer and come to understand function to see what you want to see under TEM. Okay, that's why when the fixation, the chemicals, we have to in this we must to determine the major factors which influence the cellular structures. Firstly, it's important of the most abundant molecule in the living cell, which is water, and its effect on the cellular molecules. Since water is the dipolar molecule, polarity of the specimens needs to be understood. And secondly, we must recognize the cellular physiology and metabolic processes are responsible for maintaining the cellular condition favors to the continuity of the molecule and the organelle structure. Some conditions which must be maintained, which is the homeostasis, includes temperature, pH of the system or the organism, and also the osmolarity. Okay, and like for preservative that we use usually is glutaraldehyde, paraformaldehyde, and these two is the common in our lab. And for glutaraldehydes, is the function is to fix the protein which or the amino acids, and it will cross linkings to the amino acids. Then for like osmium, it will fix the lipids part. Okay, then the post-fixation, which is we use the compound of heavy metals and give contrast, like we use the osmium tetroxide. Next is the dehydration, where to gradually replace all the water normally found in the biological tissue with a solvent, then replace it with the resin. Like for the almost most of specimen, we use either ethanol or acetone. Next is the infiltration and embedded. Infiltration is how we get the resin into the tissue or into the specimen. where the hardened resin embedded the tissue give its strength and flexibility for cutting ultracutting section. In the next of part is the sectioning, which is using ultra microtome. And then, like for the stain specimen to be viewed as DM, it must all the dehydrated, infiltrate or embedded, and also we need to undergo the sectioning for the ultra thin slices with a glass or the diamond knife. And the sections must be ultra thin so that the electron can pass through the specimens. And then usually the thickness is around 90 nanometers to 100 nanometers. It depending on how much the kV that available in the microscope. Okay. Next is for SEM, to preserve the surface of the specimen, we have to dry the sample, which is the process is critical point drying. Usually the CPD takes out the extracellular water without removing the intracellular water of the samples. And carbon dioxide, CO2, is used as a transitional fluid for during the CPD. Okay, that's the CPD. Next is the coating. Coating, we have to coat the specimen for SEM to increase the specimen conductivity. And it also enhance beam and specimen interaction, and also to avoid charging effects. Next, for negative staining is another method, which is it will not undergoes the all the dehydration, infiltration, and other for SEM. But otherwise, it use only direct staining, which is to stain the background, not directly on the sample. Okay, so in this technique, the particulate components are examined directly on a TM specimen support grid after they have been surrounded or embedded in the heavy electron dense stain, such as photostatic acid, ammonium molybdate, and also uranyl acetate. Okay, next we come into the electron microscope. So the first electron microscope is the transmission electron microscope. In short, is TEM. So usually for TEM, the specimen are fixed on the support grid, resulted either embedded in the resin or stained with the heavy metals compound, which is just now I mentioned, which negative staining. So for the copper grids, usually is very small, is around 3 diameter, and very thin, is typically less than 500 nanometer. So that's the real actual photo of how small the grid for TEM. Okay, so in our unit we have in EM is broad variety of specimen holder. So specimen holder is where we put the copper grid that has a section embedded sections, and then we put in the that hole here is the specimen holder. And but in our unit, we only have two types of specimen holder, which is a single tip, and another one is for special for 3D tomography. In TEM, we can see varies in contrast cause electrons to be dense or sparse. Certain structure within the specimen will take up more the stain than other parts of the specimen, and those specimens are called electrons dense regions. So when the electron pass through the electron dense region, they lose more energy compared to when they pass through the sparser region. And this phenomenon is used in TEM to visualize the specimen at high level of resolution and magnifications. Okay, so these are the components in TEM. So usually when the specimen is secured in the sample holder, an electron beam is generated through the electron source and shot out the electron guns, and the electron beam is accelerated down the column of the microscope after passing through the anode. And then it will go to the objective lens, and once again it lose the energy, and in objective lens it will magnify more the image before it's transferred to the projector lens, then the fluorescent screen. And this is where we start translating the electron into an image. For the image projection, The fluorescent screen is coated with a chemical which is appear as bright spots when electrons come into contact with the screen, the fluorescent screen. So the intensity of the bright spots depend on how much energy electrons have when they hit the fluorescent screen. So there's a camera under the fluorescent screen. The camera under the fluorescent screen will capture the image made from the bright spot which is displayed on the computer screen. Okay, then next is the second electron microscope, which is the scanning electron microscope, SEM. So SEM is a powerful tool which provides high magnification and high resolution three-dimensional images. It provides us topographical, morphological, and also compositional information which cannot be done in light microscope. Okay, with SEM, specimens are coated with vaporized gold or platinum ions, and because when the electrons come into contact with this coating, it causes the atoms of the surface of the specimen to emit the electrons, and these emitted electrons are called as the secondary electrons, and these are what SEM use to visualize their specimen. Okay, so this is secondary electron, where secondary electron shows the real surface of sample. It doesn't show compositional contrast, and it has sharper edges, ultrastructure. Okay, so how does the SEM work? So basically, it works similarly with light TEM. So, but in SEM, like this one is the process. High voltage is applied to the electron guns, and then the electron guns are generated, and through the anode is used to attract and fire electrons. Then the electron sample interaction, where the electrons are directed by the lenses, where electron hit the sample surface and the signal is generated. Okay, and then in the SEM, the electron beam then sweeps or scans the surface of the specimen, which causes it to discharge the secondary electron. And the detectors convert the electron signal into light signal that amplified by passing through a photo multiplier before the final image is projected on the computer screen. Then the image is generated. So these are the comparison between the TEM and SEM on how the generation of the image. Okay, next. There are two types of detectors in the sample chamber. For this is the mechanism outline diagram for the SEM. So the first detector is the secondary electron detector, and also the backscatter electron detector. Okay, for the backscatter electron detector BSE, usually it come from the deeper regions of the sample that are result of the elastic interaction between the beam and the sample. As the result from the detector are primarily used to look at the crystallography and the magnetic field of the sample. But then SEM is primarily used to study topography of cells and other similar structures. So with this type of microscopy, researchers are able to investigate the depth of the structures. Then again, for in mostly our studies for SEM is to study the topography. So this is a part of my project where this is the normal strain of E. coli. And then there are some similarities and differences of TEM and SEM. Like we know that TEM is produce two-dimensional images, like a flat image, opposed to the 3D image from the SEM. And also TEM has a higher resolution, like 120 kb that we have in our unit. However, SEM has a lower resolution, up to only 30 kilovolt. Okay, however, the main drawback of the electron microscope is it cannot visualize living specimens. So meaning all the samples must be fixed. But then the fixation will apply like through condition of the specimens. And then, of course, it's very expensive of both the electron microscope, and also it requires extensive training because of the complicated techniques to use in sample preparation for the specimen. And like for the it also requires training because it's not likely to be used like apart from the those pursuing like microbiology career. So it needs to be have a like special course for the electron microscope. And again, in summary, with EM, it's allow us to study deeper understanding on nanostructures. So these are examples that the study that we done. We study about the disrupted of myelin in human skin, multiple sclerosis. We can study on how the dynamics of the endoplasmatic reticulum, viruses, nerve cell, and other part that I think covered by our first speaker just now. And I guess that's all we know about electron microscopy. Thank you. I pass back to our moderator.
Thank you, Nur Afrina, for the insightful presentations. Before we begin to Q&A sessions, we would like to present some video demonstrating sample preparations and sample sectioning process. It worth noting that a well-executed sample processing method directly impacts the quality of the samples. This video specially for tissue processing. Different sample will have a different techniques in preparing the samples. However, the baseline still accordance to the basic sample preparations in electron microscopy. Let's enjoy the video now. We will receive the sample in glutaraldehyde. The sample will fix in GA. Once we receive, we discard the GA. And then we wash the specimen with milik water repeatedly for 6 times. After we've done the washing process, we proceed to osmium tetroxide, which is osmification process. We add osmium into the specimen. And we leave the specimen on the room temperature for 1 to 2 hours. Once done, we discard the osmium from the specimen and we wash it again using milik water for repeatedly 6 times. And then we proceed to dehydration process, where we started from 70% acetone, 90% acetone, and lastly with 100% acetone for 5 minutes each. And lastly, we remove the 100% acetone. We add the prepared EPON into the specimen and leave it for 1 hour at room temperature. And then we transfer the sample using the filter paper and transfer to the beam capsule slowly. And then we top up the EPON into the beam capsule and degas the sample in the embedding oven for half an hour, and then we spin the sample to make sure the sample stay on the middle of the beam capsule. And lastly, we top up the pure resin and label the sample and place it in the embedding oven for overnight. And this is the outcome of the sample. Then now we proceed to sectioning. The first process in the sectioning is we trim the sample in a trapezoid shape. And then we place the sample on a sample holder, and then we proceed to glass knife. Using the hand wheel, we slowly cut the sample specimen using the glass knife. Once we get the good section, we take it out the section from the glass knife and transfer to the microscopic slide. This process is to confirm the sample neat. And now it's ready to view under microscope. and this is the outcome. Once we confirm the sample, we proceed to diamond knife. We fill milled water into the diamond knife and we proceed to cut the section. We should get the perfect ribbon section. Once we get the perfect ribbon section, it will be easy for us to separate the section. It's either 2 or 3 sections. And then using the loop, we fish the sections and transfer to the copper grade slowly. And make sure the section should be in the middle of the copper grid. And for watching the videos, we hope you gain knowledge and gain insight into the work being conducted in our electron microscopy unit. We will now open for Q&A sessions. Okay, our first question is from Ms. Ratna to Puan Tengku Rogayah. What do you see as the main challenge and future directions in utilizing electron microscopy and advanced technologies such as whole genome sequencing together?
Thank you, Ms. Shalini, and thank you, Ms. Ratna. Okay, what do you see as the main challenge and future directions in utilizing electron microscopy? Okay, I will take the example of the drug discovery and also the vaccine development. I think that is the most we call that widely research done in recently. Okay, in EM. In EM, there is a new advanced technology is in the line, an EM line, which is the cryo EM, where in this cryo EM, it's allowed to visualize imaging the frozen samples in the what we call that, in the frozen proteins in the sample itself. So it was a very huge what we call the discovery in this era, where it can pair with the advanced technology in molecular technology in a molecular technique. So it is good if these two technology concurrently being used in research. Okay, thank you.
Thank you, Puan Tengku Rogayah. Our second question, they asked for Puan Nur Afrina, also from Ms. Ratna. I want to know more about cryo-electron microscopy and scanning electron microscopy in their applications to fungal pathogens research.
Okay, thank you, Ms. Ratna, for the questions. So basically, now about the cryo-electron microscope and ICM. So actually, in Malaysia, for the cryo-electron microscope, still not available yet because due to the very expensive machines, and also the maintenance will take up like a very huge budget to have the cryo. But then in cryo-electron microscope, because all from the sample prep, initially at the beginning we have to be in the cryo conditions, in terms of in the, even though in the sample prep all the fixations until dehydration, embedded, and also sectioning must be in cryo. Even for the section ultramicrotome, we need to have another type of ultramicrotome, which is the cryo ultramicrotome. And also for the electron microscope itself, also we need to have the cryo EM. So for the cryo EM, actually is very latest advancement in EM, because it we can study the structural nano particles of the specimen up to level of the protein structure, and also the structural building of the specimen itself. And also for the SEM in the application for fungal pathogens, maybe we can see how's the morphology or also the topographical image of the fungal pathogen research that can be seen into the higher level of details for and also the resolution. And also I think for fungus we can see the how the interaction and how the mechanism also, and then if is there any treatment for the fungal that we can study, and we can see how's the all the interaction of the mechanism. Okay, I guess I think I hope it's answered the questions.
Thank you, Nur Afrina. Our next question, I'll ask Puan Tengku Rogayah. From Ms. Atika, apart from TEM and SEM, is there any other type of electron microscope?
Okay, as I mentioned just now, apart from TEM, SEM, the most advanced, the latest in the EM line is cryo-EM. It's another type of EM, and we also have a reflective electron microscope. And I think in the material sciences or engineering part, they have more types of electron microscopy also.
Thank you, Puan Tengku Rogayah. Our next question is from Miss Sarah to Puan Nur Afrina. What kind of a maintenance needs to be done routinely for EM?
Okay, so when it comes to thank you the questions, Sarah. So when it comes to the machine, of course, the maintenance is needed, and also where the machine is very expensive, so it requires a very high maintenance. So usually the operator up to like in the user, we responsible in adjusting the variable bias recognition in terms of the resolution inside the machine, and we have to know to ensure that the beam of electron need to be always aligned in the column of the microscope, and also like the in terms of the camera need to be adjusted and also optimized every day, every single specimen. And also there's some of the astigmatism and more to the what the all the theory that related to the lens and the optical issues. And also like is if we have the anti-contaminators that put inside the chamber of the microscope, that one also need to be ensured every day, daily, every single day, that before using the TM. But in terms of the maintenance up to the repairing, usually it we have a subsidiary, like we have our own engineer that's related to the company, and also like they responsible for the gun source replacement, also the filament exchange and etc. And also other parts of the accessories of the EM need to be counted uh, and observed. And also the maintenance usually it takes like we have twice maintenance for a year. That one is the major like ppm. Okay, think that's thank you.
Thank you, Puan Nur Afrina. Our next questions to Puan Tengku Rogayah. What advice do you have for the beginners in the field of electron microscopy?
Okay, thank you for the questions, Miss Nurul Ain. Okay, what advice? Okay, as a I will take myself for example. Okay, what advice? In order for you to involve with this EM technology, you must understand very well the processing part of the samples, the every step of the processing part. understand the basic principle of each technique, and also you must understand the handling of the specimen, as in that's the most serious, because of course the equipment was very expensive. And then you have to understand every knobs of the electron microscopy. I don't want to be scare you all, but this is based on my experience. Okay, you have to understand the handling part of the equipment, because in EM, every type of sample is different. Is customized. The processing, the handling of the equipment, everything is customized to the sample type, and in the same sample type also is customized to the condition of the sample, whether how we as what the second present extensive trainings. Thank you.
Thank you, Puan Tengku Rogayah. By the way, my next questions to Miss Puan Nur Afrina from Miss Christina. How to handle the challenges of sample preparations for delicate or unstable materials?
Okay. Thank you for the question, Miss Christina. Okay. So actually until now, even though I've been like I think I can say 4 years lah in this field. So usually the challenges that we face, especially in the fixation, as I mentioned that in EM the sample preparation is the crucial. It's very important because in the fixation, when the fixation is done properly, then until the end, we hope that it turns out the great result for the images. That's why since for EM we receive a lot type of samples, like what Mr. Rogayah mentioned, type of the sample that we receive. That's why when we receive the sample and we have to study how's the condition of the sample first and what to look for, what's the objective that we want to see from the sample, and then what the target area. So all those things we need to sit down and consider first before we start prepare our samples. So usually the first is the fixation, because whatever because we want, as the fixation is, we want to see as much as the true condition of the specimen as in living specimens. So that's why we have to put consideration about the pH of the sample, all the homeostasis conditions, and also the temperature, and the what's the compound of the specimen. Thank you.
Thank you, Puan Nur Afrina. And then we receive one more questions from Miss Shazana Rifam to Puan Tengku Rogayah. What type of samples can be sent for scanning using EM?
Thank you for the questions. Okay, for the type of sample, as I mentioned earlier, in EM, the technology was very versatile. So we receive different type of sample. So if the sample that we never receive in our unit, but with a very strong objective, you still can send. We can explore the processings together. Okay, and one more things, the limitation also at the if you want to with sample to the scanning EM, the limitation is the what you call that, the for the microanalysis part. So but our sample usually for biological samples, not really for samples materials. So but you can send that to us, and then we can explore together. Thank you.
Thank you, Puan Tengku Rogayah. Our next questions to Puan Nur Afrina from Miss Ashura. How does the thickness affects the imaging quality?
Okay, thank you for the question, Ms. Ashura. So yes, the thickness does affect the imaging quality since it's depending on how much the kilovolt, how much strong is the gun source that available in the electron microscope that you have. Like in ours, we only the maximum is up to 120 kilovolt. Some other equipments they have up to 300 kilovolt. So if the thickness is up to 300 kilovolt, it can the thickness of the section can be more than 120 nanometers. So like ours, the application we set the requirement the requires thickness is up to 90 to 110 nanometers, because if the with our 120 kilovolt machines and the thickness of the section is more than 120 nanometers, so the electron cannot pass through the specimen, so we couldn't acquire the image and couldn't see anything because of the electron is not enough to generate the image. That's all. Thank you.
Thank you, Puan Nur Afrina. And I hope this will be the last questions from Miss Aina to Puan Tengku Rogayah. Where else have EM facilities other than IMR?
Okay, under Ministry of Health, Makmal Kesihatan Awam Sungai Buloh also have these facilities, but with different objective and different functions like us. And most of the public universities such as UPM, UKM also have these facilities. Thank you.
Thank you, Puan Tengku Rogayah. Well, we have reached the end of our question and answer sessions. Any additional questions will be addressed by the speakers via emails. We hope this webinar has illuminated some of the research work being conducted at IMR and that it has sparked interest, potentially leading to future research collaboration with us. Feel free to contact our officers for any research collaboration courses or any lab visits via their email which mentioned in the screen. We have reached the end of the sessions. I would like to thank all the participants for your supports in making this session a success. Please don't forget to subscribe to our National Institute of Health YouTube channels. Before I conclude, I am pleased to announce the next sessions of the IMR-SEAMEO Webinar series scheduled for August 30th, 2024. Mark your calendars. The theme for the next webinar is Oral Cancer in Focus, Bridging the Clinical and Histopathological Perspectives Towards an Integrated Patient Care. Until then, many thanks from all of us at IMR. Take care and stay safe. Bye-bye.